We will continue with our next presenter. Lukas Widmer is a statistical consultant at Novartis, where he collaborates with cross-functional teams to further develop quantitative methodology in global drug development. In this presentation, Lukas Widmer will show a new drug-drug interaction model, and I hear he's joining us remotely. Hello, Lukas.
Hello. I'm Lukas Widmer from Advanced Exploratory Analytics at Novartis. And over the next 15 minutes, I'll give you a peek into data science in pharma, specifically into safely guiding multi-drug dose escalation. First, I'd like to state that the opinions expressed in this presentation and on the following slides are solely of the presenter and not necessarily those of Novartis. With that out of the way, I'd like to turn your focus to the biopharmaceutical research and development process. So we start out in basic research, and as a drug evolves through drug discovery and then preclinical testing, so basically lab-based testing, there comes a point where we go into our first human trials. And typically this is in human volunteers, so phase 1 trials. And after that, hopefully the drug will evolve through phase 2 and 3 trials and eventually go through review and hopefully be approved and then go to market. In this talk, we'll focus on phase 1 trials, which are the first trials in human volunteers. Specifically, I'm gonna give you a bit of an overview of phase 1 trials in oncology and why acquiring big data there is really not an option. First of all, volunteers there are typically patients, and these patients have cancers that have progressed despite standard treatments. So not any data can be collected. There's some ethical and operational challenges to this. First of all, we have to maximize efficacy just due to the urgent need to have a treatment effect. This is due to the life-threatening nature of the disease. One must not avoid giving knowingly working treatments to patients in order to study a new drug. This is also ethics 101. And then we also need to manage toxicity risk to patients due to overdosing. So we must control this given limited data. And last but not least, there are not so many patients that actually qualify into these criteria. So in this slide, you see why we have to strike a delicate balance between sub-therapeutic and overly toxic dosing. You see an x-axis that starts at 0 dose and goes to high dose. And at 0 dose, obviously, we're at sub-therapeutic dosing, where lack of efficacy would lead to death of the patient due to cancer. And on the other end, it's likely that one would find serious side effects or toxicity. So the probability of a dose-limiting toxicity, DLT event, is high in that region. So the goal of these dose escalation studies is to systematically increase the dosing as quickly as possible, so we reach a biologically active and hopefully efficacious dose, but also to do this as safely as possible such that these DLT events are controlled. And the final goal then is to determine the final dose, the maximum tolerated dose, or MTD. Now, initially, we have quite limited knowledge on toxicity for a given dose because we come out of preclinical testing, and this is the first time we go into humans. And then even if we could recruit a large sample of patients initially, this would be unethical. So what do we do instead? Instead, we start with a small sample and perform adaptive escalation with overdose control, so-called EWOC, step by step to warrant patient safety. To do that, we employ a Bayesian logistic regression model, henceforth BLRM, that models the outcome of one cohort, so 3 to 6 patients usually in this case, with a probability of generating a dose-limiting event pi. This just has an odds of alpha when we're at this reference dose dref, and a constraint slope beta that is greater than 0 that ensures that toxicity increases with increasing dose. This DLT probability we actually stratify into 3 regions. The first one is actually underdosing, so between 0 and 16% of DLT events probability, we actually would be we would accept living with sort of higher toxicity given the lethality of the disease. Between 16 and 33% were in the target dose region, and above that, that will be overdosing. Now, EVOC for a specific dose, D, is fulfilled if the probability of being in this overdosing interval, so the mass, is smaller than 25%. Now, armed with this EVOC criterion, what we do is encode all of our preclinical knowledge, historical and trial data, and I'll get into that shortly, into this BLRM. And for the doses that you can see on the right side, determine which ones of these, given this prior, fulfill EVOC, and those can then be used to enroll patients. So we would start, for example, at the starting dose indicated in this black rectangle, and enroll 3 to 6 patients into this dose level, observe how many DLT events we get, and then basically update the model. And we'd be updating the model after each cohort until we reach the maximum tolerated dose. Now, obviously, this is quite sparse data. 3 to 6 patients is not a lot, but we can actually really try to make the most of this sparse cohort data. And the way we do that is by also considering historical and concurrent data, both of which are sort of termed co-data. And the reason why this is a good idea is that, for example, the drug might have been tested for another indication in a slightly different population where we already have data, so historical data. Or then in the case of newer drugs these days, we can actually usually combine more of them together because they tend to be less toxic. So the MTD is rarely reached by a single drug. And then if you have a trial with many simultaneous drugs, for example, a double combination backbone A plus B and multiple triplets A plus B plus C, A plus B plus D, the triplet can of course learn from an ongoing backbone trial. So if you'd have a trial covering drugs A plus B plus C, you'd obviously want to integrate data from drugs A and B as well. And the way we do that in a robust manner, so borrow information from co-data in a robust manner, is by XNex, the principle of exchangeability, non-exchangeability, where first of all we have a big model, this exchangeable model that takes into account heterogeneity between groups, where groups are, for example, historical trials or concurrent trial arms. And information is borrowed between these exchangeable groups governed by heterogeneity parameter tau. And basically, the higher tau gets, the less data you would be borrowing between the trials because the between trial variability is higher. On top of that, we model each group as a mixture of an exchangeable and non-exchangeable model. And this is really there to facilitate borrowing of co-data that is unobserved at the design stage. So you have to build in robustness at the design stage to take into account co-data that might be coming from a different distribution. And this is actually equivalent to Bayesian model averaging of a model with hierarchical parameter modeling, so the big one, and a model with stratified parameter estimation by group. On top of that, we need to model drug-drug interaction safety. And I won't go into the full mathematical detail there, but it's open source, so you can look into it after the talk if you're interested. But basically, there's 2 desirable properties that we want to enforce. The first one is just consistency. If any of the drugs goes to dose 0, the interaction term should cancel, and the model should reduce to the marginal. On top of that, we also want to ensure that asymptotically, the DLT probability goes to 1 if any dose goes to infinity. And that's actually something that our proposed model improves over the state of the art, which only fulfills A. The reason why this is a good idea actually goes back to Paracelsus, who once stated that all things are poison and nothing is without poison. The dosage alone makes it so a thing is not a poison. Even water will become toxic at very high doses. So to give you an intuition on the effect this has, what you see here is the current model on the left, the proposed model on the right, and the top is the plots for EVOC failure. So the red region is forbidden, the blue region you can run at, and on the bottom you see the probability of overdosing, and X and Y axes are just dose, increasing dose for the 2 drugs, A and B. And as you can see on the right, bottom right, where the arrow is, the overdose probability now asymptotically only increases with the proposed model and not with the current one. Now you might ask as to what impact such a subtle change has. And to determine that, we benchmark against clinical trials with known truths. So we generate a grand truth. Here we run an example trial with 4 drugs, A through D, and 4 arms. So we start with A plus B, and then open up arms A plus B plus C, the same but with a drug holiday, and A plus B plus D. And then sample a virtual patient population with a known grand truth that we specify, where each dose has a known probability of generating a dose-limiting toxicity event in a patient, and we simulate cohorts according to the respective binomial distribution with 3 to 6 patients. Finally, we benchmark both models against an oracle that escalates provably optimally and uses grand truth data to escalate optimally. So what grand truth data, you might ask? We're going to benchmark 3 grand truths here. The first one being that no dose combinations are toxic, which is a sanity check. Escalation should there go as high as possible, as fast as possible, so we can optimally treat the cancer. Then second of all, we'll run a typical trial, so sort of what a trial statistician would expect. And the third scenario is basically the same, but with 1 toxic arm. That acts as a stress test for the interaction model. This would be A plus B plus D in this case. Now, without further ado, let's get to some results. So the 2 first criteria we're going to consider here, which are also the most important, are the risk to current and future patients, where the risk to the current patients would be defined as the probability of being put on a toxic dose in the dose escalation trial itself. And the risk to future patients would be the probability of declaring an MTD that is in this toxic range. And as you can see, the oracle, of course, escalates optimally, so the probability is strictly 0. But as you can also see, this quite subtle change we made here actually resulted in 8% fewer simulated patients to be on a toxic dose, so the risk to current patients is lower, and 13% fewer simulated trial arms declared a toxic MTD, so the risk to future patients is also lower. Then we also come to trial cost and speed. This we measure by the expected sample size per arm, so expected number of patients per arm, and the expected number of cohorts to finish a trial arm. And again, here the oracle is obviously the fastest. And over the 12,000 trial arms you see here, we actually required around 7,000 less patients in our proposal, so 3% fewer patients, but it's actually 17% closer to optimal. And we're also faster. So we actually need 2% fewer cohorts, and which is this is 21% closer to optimal. So that already brings me to the conclusion. What I showed you is that we encode preclinical, historical, and domain knowledge into a robust prior that gets updated with sparse data from every cohort. And this actually allows us to limit patients' overdosing risk while escalating to a therapeutic dose quickly. This response surface modeling with asymptotic properties defined by domain expertise really allows for enrolling fewer patients for dose finding, can accelerate clinical trial success, and most importantly, controls for toxic dosing better. If you're interested, you can try simulating your own dose escalation trial with our open source Oncobase 2R package at the following URL. Or a QR code where the math is also extensively documented. And with that, I'd like to thank the organizing committee for inviting me, for your attention, and we're open for questions.
Thank you, Lucas. And we got a couple of questions. For example, how do you deal with external factors such as gender, age, ethnicity? Does this not make it very hard to make general predictions?
Yes, actually, that's something I sort of hit when I made the argument that the population can be different, right? And this you basically need to encode into your prior. So, if, for example, if you have historical data from a trial where the distribution in these covariates was different, you need to make up your mind what your belief is and how close this could be, and also what the probability of this really departing from your new trial could be. So this is setting these X and X weights, and that is how you account for that. It's really encoding this into the prior.
Did you consider survival analysis for drug toxicity being a trigger for treatment change escalation?
So I'm not exactly sure what treatment exchange. Could you repeat the last part of the question?
Yes.
Treatment exchange escalation?
Sure. Yeah, being a trigger for treatment change / escalation.
Okay. So in terms of survival analysis, you can also go deeper and start looking at, for example, time to event, so time to toxicity event. And depending on your trial design, you could also, for example, think about, okay, are we at some point going to include a decision point where we would change the treatment entirely? But that would be sort of out of scope for this analysis.
Okay. As a potential patient, wouldn't it be safer if Novartis would actually collect more data instead of inferring the probability as you suggested?
So there's a couple, this is a really good question. There's a couple of challenges to this. I mean, the patients that we're looking at are patients whose cancers really progressed despite the standard treatments. So this group is already not that big because the disease is actually quite severe. So I mean, that's one major challenge. The other is that initially, if you're uncertain of the toxicity of your drug because you haven't tested in humans yet, this, of course, isn't a good idea to immediately acquire a lot of data. I mean, if I could pull up slide 3 again, obviously there's going to be further clinical trials down the line. We're talking about the first trials in human volunteers, right? We are not talking about later phases where we'd obviously have in a confirmatory setting many more patients.
Okay.
So that's a threefold answer to this question.
Thank you. Maybe one final question. Can the method be applied to other data than oncology, and if yes, for which?
Yes, of course. This can be applied to other data. I mean, in this case, you'd probably not want to use the OncoBase-2 package, but look into Bayesian evidence synthesis. So Sebastian Weber's R package that is also open source and on CRAN. And yeah, I would suggest you look into that.
Thank you very much, Lucas. And I'm afraid we have to continue here with the program, but thank you very much for your time.
Thanks a lot, and I'll tune in.